Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. Today's guests are the co-founders of GeneMode, Jacob Lee and Jin Cho. Jacob and Jin met in ninth grade English class. GeneMode was born out of a need that Jacob identified while working in the lab. What is GeneMode, you ask? GeneMode is a platform that aims to help scientists manage their research project more efficiently, and its mission is to make research repeatable and reproducible to find cures to diseases faster. Before jumping into this episode, I want to take a moment to tell you that this week is the Dr. GPCR Summit Week. We have over 30 speakers, 360 registrations from all over the world, and I hope you're able to join us. It's not too late to attend. Just visit drgpcr.com/summit-2020 to register. And now, let's dive into our episode with GeneMode co-founders Jacob and Jin. Hello, everybody. I'm Dr. Yamina Bershish, founder of Dr. GPCR. On this very special episode of the Dr. GPCR podcast, we have two very fantastic guests, Jacob Lee and Jin Cho. They are the founders and co-founders of a company called GeneMode, and we'll talk a little bit more about the company, but I wanted to introduce you to our founders. Hi, guys.
Hey, Amina.
Hey, Amina.
How are you guys?
For having us here.
Of course. Thank you for being here, both of you.
Cool. So I'm happy to, talk a little bit about myself. So I'm Jacob, CEO, co-founder at GeneMode. And GeneMode is a software company in the life sciences and lab management space that simplifies research for scientists. A lot of the problems that we're solving is around data sharing for scientists. By having inventory, experiment management, applications, data, all in one collaborative workspace. Our goal is to be the gold standard platform that accelerates research for scientists so that research is repeatable and cures are developed faster.
That's awesome. And hi, Jin. How are you?
Good, how are you?
Good. So you're the co-founder of GeneMode. What do you do at GeneMode?
Yeah, I guess let me, introduce myself. So, my name is Jin. I'm CTO and co-founder of GeneMode. And I guess, ever since I was young, I've always enjoyed working with technology, and I studied electrical computer engineering in college. And after I got my first job in the defense industry for a company called General Atomics, where we built software for military drones, I picked up a lot of skills, especially in software side, and they really helped me optimize the workflows for research scientists that I'm building in GeneMode.
That's awesome. And Jacob, you have a scientific background. Can you tell us a little bit more about it?
Yeah, sure. My background is in bioengineering, where I worked as a scientist in stem cell research and cancer research. So these experiences helped me understand two things. First, I was surprised to learn that research technology is still stuck in the 20th century. And second, I realized that research, although very important, is just a very small piece of the puzzle. So, these two realizations helped me spark a desire in me to do more and to make a bigger impact, not only in the research community, but also for patients who depend on drugs and vaccines to improve their health. And my way of making this impact was by starting a company that simplifies and speeds up that research process so that patients can get drugs and vaccines faster.
Awesome. And where did you two guys meet?
Yeah, I can talk over that a little bit. So our history actually goes way back to our ninth grade English class in high school.
Oh,
And
wow.
and since then, we became close friends, and we went to the same college, and we kept in touch even after we studied different majors and took different career paths, and we kind of reunited for GMUD after that.
Awesome. Can you tell me more? How did you come up with the idea of working together at the company and founding the company? Was it, I don't know, some kind of an cool story around a beer, or some having a discussion about some frustrations that Jacob you had mentioned around working in the lab? Can you tell me more about that?
Yeah, definitely. So I guess after college, both Jacob and I were working um, in industry. When Jacob was working as a scientist, he shared his experience of using like legacy software and how difficult it was to learn and complicated to use. So I looked into it and I was very surprised. It looks like a software that was built a couple of decades ago. And like what surprised me even more was his lab was paying thousands of dollars for this legacy looking software. Like I was mind blown, and like this was the opportunity where I could use my um the modern technology to improve the experience for scientists who are still living in like the last decade.
Awesome. And Jacob, what were some of these frustrations you had when you were using these antiquated softwares?
Yeah, so I mean, just to echo Jin, this idea came at the end of my first year in grad school, and at that time I was working in that research lab. Was using that expensive software that looked something straight out of Windows XP. So my frustration was that in a lab of 10 people, only the principal investigator and a very senior postdoc had access to the software. And as a student, I had to actually schedule times on my postdoc's calendar in 30-minute segments even to learn how to use that tool. And so that's when I thought, hey, this, isn't fair. It doesn't really make sense that, this software accessibility issue since 2018. And so that's, when I messaged my good friend Jin, who I know is always up to date with the latest technology, and asked them, hey, is there something we can hack together and bring, scientific software to the 21st century? That's really when we built our first MVP, to fit into the way that scientists are already doing their research. And we grew our company from there. It's been super exciting journey.
That is awesome. And tell us more about what can GeneMod do that, for example, an Excel sheet or that antiquated software can't do.
Yeah, definitely. I can talk about it a little bit. So I guess the biggest difference between like Gmod and like Excel or those antiquated software is that Gmod was built for life scientists, whereas Excel wasn't. And our platform has an interface that automates a lot of data entry processes. So imagine like putting the information like your name in Excel. Like what you have to do is like you type for every cell like your name or copy paste for each individual cell. Whereas Gmod, it auto populates those names, dates, and some of the essential scientific information. It really reduces the time for research scientists to enter their data. I guess another thing that makes Gmod different is that it's really easy for research scientists to collaborate on their data. If you have used Excel or SharePoint before, like you might know the pain of the file being locked by someone else that you always have to ping the person or wait until it's released.
Yeah.
And like our platform, solves this issue by providing a real-time editing and like notification system
Mhm.
so that when things are changed by others, um, every user get instant feedback on the platform.
That's awesome. I have to admit, I did try out GeneMod and I really enjoyed how easy and how intuitive it was to enter information and to navigate the platform. And that's why I wanted to talk to you guys so that you can tell us more about your story and help spread the word about how fantastic of a product it is, at least in my opinion, and in my limited time that I've tested it out. Who is this product for? Is it for life scientists in academic environment, or is it for scientists in any environment as long as they're life scientists?
Yeah, I can tackle that. It's for any scientist. So it doesn't. You don't have to be in a specific field like biochemistry or immunology to use it. We a lot of our labs that are on our platform come from many different backgrounds. In fact, a lot of the labs at Fred Hutch, Cheetah Medicine, Seattle Children's, they all see that same benefit regardless of their fields, helping them reduce that amount of money that's spent on unnecessary reagents, repurchasing, helping them streamline their data management, improving that team collaboration.
Awesome. Awesome. Can you give us a specific example of how a bench scientist, a life scientist, would take advantage of using the software? For example, I want to sign up today to try it out. What do I do? How do I do that?
Yeah, for sure. Real simply, our website is www.gmod.net. That's GeneMod. Feel free to check us out there and give our platform a try. We're always happy to set up scientists, new labs to work into their workflows. Help them onboard into this aspect of lab automation, increased reproducibility. Just to kind of go to an earlier point, really the problem with traditional inventory management tools is the variability in how that data is being recorded, right? So in a lab of 10 people, each person might have their own spreadsheet, and that creates a lot of confusion. You might have to ask people for their Excel spreadsheets, or you might not know if this is up to date. So GeneMode helps reduce that variability and that data capture. And by having everyone on one platform, it fosters that more of that collaboration, that transparency that's needed.
I think that's essential. I oftentimes get emails from previous colleagues asking me for my Excel sheet with, like, for example, my reagents or my constructs, and I've certainly been in a situation where the freezer was so old and so frozen that nobody was venturing into opening that freezer and figuring out what tubes were in there. It was much easier to go ahead and order, and having a product like yours would help everyone track what inventory there is available in the lab, but also go back hopefully decades and figure out who did what, and what's in that particular box. So the other question I had for you is, how do you see this product, your product, help advance scientific discoveries? I think you touched a little bit on it, but I wanted to know if you could elaborate a little bit more.
Yeah, our platform is also much more than a freezer management platform. It also is a project management platform. So this allows the entire research team to be on the same page about their samples, experiments, and timelines. And this is also a huge differentiating factor from Excel sheets, which is why our platform is useful not only to inventory management, but to the entire drug and vaccine development process. In fact, each year, 10 billion is wasted on unnecessary purchasing of reagents in the United States. And that's a massive resource sink just for that pure error and variability that's sourced from Excel. And so, to that end, a lot of the work that we want to do is to make that more efficient, to develop cures faster so that we can deliver that to the bedside.
Obviously, and it's an important component in drug discovery and in research in general to be organized and to figure out what is where. And I think it would help very much keep everybody organized and advance research and ultimately improve human health. So you had mentioned where people can find you. What's the best way to get in touch with you? Through your website? And what kind of minimal information do you require to have users try your platform?
We want to simplify the onboarding process as easy as possible. And in fact, that was one of our early missions starting GeneMode is, you don't really have to read a lengthy manual, a how-to guide. You can get started right away because, we envision this as something that fits into what people are already doing, how they already do their research. So we want to make that a seamless fit. I think, the best way would be to check out our website, look at some of the demos that we have there, some of the great blogs and case studies, and then request access. And, we should be in touch within a day and we'll get you set up.
That's so great. Who came up with the name? And does it mean anything?
So it was kind of a joint effort. It was in our ideation phase, and it's kind of split into two. You can think about it as GeneMode. Actually, it's shorthand for a genome modification. We started out actually building a tool that was a very simple tool just to help with some sequencing. Obviously, the problem is so much bigger than that. And as we worked on GeneMode, we discovered that, the scope of the problem was much larger. But since then, we still retain to the old legacy name.
Awesome. Awesome. Did you have other name variations that you may or may not be proud of, and you're happy that you got away from those?
I can't recall too much, but I do know that at some point we probably had a list of 20, 30 names we were choosing from, and we ended up on something that was genome related. We wanted to kind of have that, scientific component in the name.
Got it. Got it. Then you in the platform has also a sequence alignment, and you can do sequence work, and you can use it in that way. So I think it's a fitting name. So you had mentioned that it's not only a freezer and um, tube management software. You can do project management on it. You can analyze sequences, look at sequences on it. What are the plans? Are you planning on integrating? If you can talk to me about it, obviously. Are you planning on integrating more tools into GeneMode?
Yeah, absolutely. In fact, we have a lot of neat ideas of tools that can benefit scientists. So, there's many different aspects to this experimental life cycle to complete a life cycle of experiment. We plan to develop tools that address this entire life cycle, from basic R&D, potentially to manufacturing, potentially to later down clinical applications, with all the data that we can provide back to the user, to the clinician, to the researcher, to help them with their goals. So we absolutely do have in mind a pretty good timeline of product releases, and for users joining on now, they'll be having that early access to also help test some of those things.
Obviously, and just remind me again. So you do rely on people's feedback. What has been the consensus on your users? What do they think about the platform?
Yeah, the overall consensus has been great. It's been super positive. In fact, a lot of our new users, new labs, come on based on organic referrals, based on hey, a colleague of mine checked out GMod. Is it possible for me to get access as well? And I give a lot of thought to this. Kind of the value that we bring. I think in one way it's that it is on the product side as well. Not only on the mission but on the product, right? We allow users to visually see where their samples are. Unlike tabs or sheets on Microsoft Excel, people have an actual virtual freezer broken down into further shelves, racks, and boxes. And then there's a host of other features, whether it's a robust search engine or other bells and whistles that we can provide.
Exactly, and if I remember correctly, when I was testing out the platform, you had mentioned that you would be able to enter an inventory, for example, of consumables, and it would track the usage of these consumables and alert you on when to order more. Can you tell us a little bit more about that aspect?
Yeah, so a lot of the algorithms that we plan to build or have already built will remove that heavy lifting work from the scientists. So a lot of that paperwork, that overhead, we can automate those processes. So let's say we see that samples or reagents are being more frequently used than others, then we can start flagging that. We can have that into alert systems. And we can have those alerts or notifications back to the user so that they know, hey, this is something that might be running low. We might have to reorder that in the future. Other extents that we can do is, for example, like projects, right? So protocols that we know require a list of reagents or a known quantity. Those are things that we can also input into our system, and so they can quickly see like, hey, do I even have enough reagents to run 5 experiments or 5 protocols, and they can forecast that accordingly.
And that's very useful. I'm sure our audience and the lab people in our audience can relate to that. When you plan on doing an experiment, you're doing it, you're in the middle of it, and then that's when you realize that there's something missing or the bottle is empty in the box in the fridge. So I think having a tool that can predict ahead of time, with X quantity of reagent, this is the number of experiments you can do, and also track the usage of each lab member, especially if it's a common reagent. That can be very useful. On a more practical level, for example, if you're a lab of 10 people, you sign up to try out and to use GeneMode. How does it look for those 10 people? Is there an admin, or everybody has kind of a democratic access to it? Do you have a private a section in the account, or everybody has access to everything? It's a lot of questions in one, so I'm happy to repeat them in case I missed something.
Yeah, sure. I can give a basic go at that. And feel free to chime in if you have anything up to add on the tech side. But at a high level, we do have a basic differentiation between admins and members. Admins of a lab, they do have more privileges, whether it's making more of those decisions, such as deleting entire freezers, deleting projects, managing permissions. So we do build those administration layers in. Other than that, there's not too much. I do imagine that as we build out the tool more, what I mean by that is having additional products. We'll have more distinction between admin and members. But for now, it's just as easy for an admin or a member to get to work, to get their research where it needs to be.
Okay. Okay. Great. Great. And then would each user, independently whether they're an admin or not, have their kind of a private section within the tool?
Yeah, so we always give the individual user, even though they're part of the same team, we still give them the ability to have their own private projects or private freezers. So if they don't want, let's just say, their mini fridge reagents or antibodies to be shared, they can create a private freezer that's just for them. More often than not, we obviously see a lot of labs, they just have everything as shared. Of course, that fosters more collaboration, but we don't take that ability away if someone does want to keep that to themselves.
Got it. Well, that's, yeah, collaboration is the key of everything, and having the ability to see across the entire team what is available and what constructs, for example, or what reagents are available is very important in that sense. Jin, anything on the technical side you'd like to add? How hard is it? How big is the team, and how do you function on a daily basis to add on these features?
Yeah, that's a good question. So our team, it's very streamlined. We have a very good infrastructure to process all these feature developments and fixing all the software bugs. So weekly, we make updates at the end of the week, and for each week, we kind of set a priority of which features do you want to develop for this week, and what are the bugs that customer reported, and what are the features the customer requested, and based on those, we kind of set the priority, and that's how I kind of hand off to our designers and engineering team, and from there, yeah, we go really rapid developing phase for first few days of the week, and then towards the end of the week, we kind of wrap up our work and do a lot of testings on our testing server, and once everything is verified, that's when we ship it to a customer and have it live on our production server. And yeah, our customers really liked about how fast we address their requests, and I think yeah, this is one of our advantages, how we can make changes weekly basis.
That is great, and I think what the best thing is to have customer reviews and customer input, because at the end of the day you want to make sure that users are happy with the product and users get what they need so that they can unburden themselves with entering information endlessly like they would do in Excel, and move forward their research. All right, so thank you both for being here today again. I wanted to ask you a quick little question regarding, this whole COVID situation. And if you could talk to us a little bit about how it changed or hasn't changed your routine, knowing that the work that you're doing is computer-based.
Yeah, I can speak to that. So COVID-19 is obviously top of mind for everyone right now. And I think the question that we ask is, why is it going to take a year and a half to get to the vaccine? Why does it take so long? And that's because the drug development process is lengthy and it has three phases: the preclinical, the clinical, and the FDA regulatory phase. So when COVID rolled around, it certainly helped us. I think we might not know how to solve COVID, but we certainly are building the tools that to provide to those researchers at the front line. And so at GMOD, the end goal is really much bigger than helping scientists manage their research projects or inventory. Our mission is to make that research repeatable and reproducible so that we can get those cures faster.
Exactly. And the best way through it is through tools that can help researchers, spare time and focus on what's important and make sure that, they can do their work and focus on making discoveries and hopefully advance and improve human health.
Right.
Super. I'm really glad that you had the time to speak to us today. I wish you all the best. As we said, so you're available online. In the transcript of this podcast, I will put both your LinkedIn profile links, but also the link to GeneMode so that people can find you, and the request to join the community and use GeneMode, hopefully to improve human health as best as possible. Was there anything else you wanted to tell us about GeneMode before we leave today?
No, I would just leave everyone with this, being at that forefront of lab automation and reproducibility is important. And we hope that by using GMOD, this is the first step in that direction for future scientific research. And, again, I want to thank you for your time. I mean, it's always a pleasure talking with you.
Thank you so much. Thank you both. Thank you, Jin. Thank you, Jacob. I wish you all the best. We'll be in touch. And this concludes this episode. I'll talk to you soon. Bye, everyone.
Bye.
Bye.
Thank you for listening to the Dr. GPCR podcast. We hope you enjoyed it. We look forward to having you attend the Dr. GPCR Summit. If you'd like to sponsor us, please visit drgpcr.com/sponsor. Thank you to GeneMode co-founders Jacob Lee and Jin Cho. Thank you to Attila Forrest. Music by Rosa Bershish. We'd like also to thank, as usual, our very dedicated and talented science communicators, Shivani Sachdev and Jin Chong. I'm your host, Dr. Yamina Bershish. Thank you for the privilege of your time, and until next time, stay safe.